All right, we're gonna get it started. Can you all hear me? These microphones working? All right. So, welcome to Cognitive Neuroscience. This is 6101. Make sure you're in the right place. We found the room, so that's good. We'll be meeting here on Tuesdays from 6 to around 8 p.m. The course is a graduate level course, as you all know, and I think there are a lot of master's students here, but we also have a few undergraduate students that had permission to take the course. We're gonna start by going through the syllabus. I'm gonna answer any questions you may have about it. And then we'll do a little lecture on neurophysiology for today. If you have any questions, please do ask. It's good that you participate in the process. It's good that you participate in the class in general. So as I said, we're meeting on Tuesdays, 6 to 7:50. I am Hossein Adeli. I'm the instructor for the course. I'm part of New School as a part-time faculty, which basically means a teaching faculty. My main affiliation is with the Zuckerman Institute uptown. My office hours are right before the class from 4:30 to 5:30. In the lounge, it's the building right across on the 7th floor. You'll find it. The TAs, we have 3 TAs for the course, Nadia, Kate, and Cami. They're sitting in the front. Nadia has a conflict, so she's not going to be in the class, but Cami and Kate will be coming every week. The thing about them is that they're very knowledgeable in this course. Kate and Cami had the course a couple of years ago, and Nadia has already TA'd for the course a few times. So they all are very familiar with the topic, and they're great resources for you all to go to if you have any questions, if you have any concerns. In general, it's your point of contact should be the TAs. We will assign students to TAs. We've already done that alphabetically. And you will get an email, or you've already gotten an email from a TA saying that, oh, I'm your TA. This is for just the division of labor, where we have a large class of 60 or so students. So it's good that we assign 20 students to each TA, and they would be the ones that would be grading your exams and your assignments. Their office hours are listed here. You could go to those office hours from your own TA, but you should also feel free to go to the office hours of other TAs if they fit your schedule better. They all know the material, so any questions you may have, they should be able to answer. As I said, you'll get an email. You've already gotten an email about it. The syllabus, I'm sharing it as a Google Doc. The reason being that we're going to add, for example, Zoom links or the schedule for exam sessions or stuff like that. So you don't want to print this out or save it. If you need to look at the syllabus, just look at the Google Doc. The course, you know what the course is. You've all seen the description. We'll try to kind of cover some of the basics of brain science in general. And we'll try to talk about cognition. So this is not a clinical psychology course. This is not a clinical neuroscience course or a social psychology course. This is a cognitive neuroscience course. So we talk about the brain and we talk about how it relates to cognition. Just make sure that you understand that, because we get occasionally comments about it. The focus of the course is cognition. So you'll learn a basic understanding of neuroanatomy, of neurophysiology. You'll become familiar with different neuroscience techniques in terms of recording brain activity. You'll hopefully, by the end of this, you'll be able to pick up any paper that is relevant to you or your research and be able to work your way through that paper. That's the idea. We want to give you a foundation for whatever you want to do next. We'll cover topics like recognition, memory, perception, attention, topics that are generally interesting in the context of cognition. We have a textbook. I'm calling it optional. The reason being, whatever you'll be asked on the exam will be covered in the class. Having said that, some of you want to go deeper on certain materials, and that's the textbook is a great resource for doing that. Some of you come from a background that is not very strong in science, and for you also, the textbook is a great resource. In general, it's a resource for you to go to if you need anything, beyond for your own learning or as it relates to the course. Whatever will be on the exam will be covered in the class. I uploaded a PDF copy of the textbook on Canvas, so you don't really have to pay for it. It's there, so you could access it. We will have 2 assignments, and the first one will be writing. You will write a reaction paper to an article. We'll give you a couple of options. You'll pick the article that you like, and you write what your understanding of it is, and you answer a few questions about it. For example, what are the main findings, what tools they use. We'll give you a template for doing that. The second assignment will be on presenting a paper. Again, we'll give you a few options. We'll pick one of them. And since there are a lot of you and we don't have enough time, the idea of this presentation would be for you to record yourself. So we'll pick the paper, you record your presentation in Zoom or other applications. I'm using Zoom, for example, here. So I'm starting a meeting, then I'm sharing the screen, then I'm recording to the device or the cloud. And then after you're done, you get a link, or the video gets saved on your computer. And then the best thing to do is to upload that on Google Drive or YouTube and then share that link and make sure the permissions are right. Again, these assignments, we'll talk about them more as we get closer to those deadlines, but I'm just giving you an overview now. Those are the 2 assignments. We have 3 exams spread out across the course. Each will be 20% of your final grade. It would be a mix of short essay questions and multiple choice questions. The short essay questions will all come from a study guide. You'll get a study guide. A subset of those will appear as short essay questions. The multiple choice questions could come from anywhere that we covered in the class. So this will encourage you to study all the material besides the study guide. We'll have a final paper. This is an opportunity for you to explore a topic further beyond what we cover in the class, some topic that you're interested in. You could pick that up, you read 5 to 7 papers on it, and then you summarize what you found. So in a way, it's a review paper. And as I said, it's an opportunity for you to get deeper on a topic that you find interesting. The formatting and the requirements are all listed here. We'll go through them as we get closer to the actual deadline of this. We do have a late submission policy. It is for your own good, in a sense that it's good to learn to be on time with the assignments. Just check it out. It says if your paper is not in the system by the time that it should be, you lose 10 points. And then for any extra day, you lose 2 more points. So do the work on time, do the work early. The grading rubric is standard new school grading rubric. We have the 100 points that I mentioned, which is 10 for each assignment, 60 for the exams, and then 20 for the final paper. That's 100. Then we'll have 3 extra points. This is a good student, quote unquote, extra point, which means that you just show up, you participate, and you take the class serious, and you do your best. You know, that's really, you all know what a good student is. So altogether, it's going to be 103 points. If it's above 93, it's an A, and then you could see the other ones. A couple of years ago, I created this tool, if you want to call it that, because this is not a simple summation. So for people to be able to calculate their grades. So you're not touching anything beyond this point. You're just putting, this was just, the intention here was for you to be able to monitor how well you're doing, depending on what kind of letter grade that you want to get. So you could put your grades in, the grades that you have up to a certain point and the grade that you would expect to get, and then you just calculate it. Basically, you run the code. This is a simple Python code. And then at the end, you get your grade. Now, if you put a 80 here, for exam 3, what happens? Yeah, you get a new grade. So again, this is just a simple thing for you to monitor how well you're doing as the semester goes on. My intention is to record all the lectures, barring any technical difficulty. I will make it available to you tonight or tomorrow morning. This does not replace you attending the class. Attendance is mandatory. We'll take attendance. So you should show up, and you should only use the recordings as an extra resource as you study for the exam. You could watch certain sections again, just to see if you didn't understand the material, or if you just want to check something. It will be made available to you. If you have the health situation, we'll follow the school policies. Final note on this section is we all use chatbots and these sorts of newer tools that are out there. There are some restrictions because this is an educational environment, and the intention here is that for you to learn certain things, some of it being developing your own voice, learning how to write, right? Any use of chatbot that goes against those objectives, then it's prohibited. That just makes sense, right? You asking a chatbot to summarize a paper for you and submitting that is clearly not the intention of this course. So use good judgment. And there are ways that you could use these systems that are acceptable. And I listed a few here. For example, you need ideas, and then you could, then research them yourself. Some of you need help on checking grammar, and these tools could do that for you. But any use should not go against the learning objective of the course. You should not submit something that is entirely generated by these systems as your own work. Try to do everything yourself. If, as I said, you need a little help from these systems for grammar or for organization or something like that, then use it, and then have a paragraph explaining how you used it, right? This is helpful for us to know, because you're going to take the exams here, pen and paper. It's going to be if it's going to be obviously different than how your paper sounds, then it's going to be alarming to us, right? We're going to see that. So if you happen to use them, then just explain how you use them, and if it's within the accepted norm, then we'll accept it. Is that clear? All right, the syllabus is organized by week. This is the first week, August 27th. We'll review the syllabus. We'll look at neuroanatomy a little neurophysiology. If there are readings or resources, they will appear on the box below. We'll have a crash course on basics of neuroanatomy and cell biology by Kate. Is this decided the time yet?
Well, I just wanted a reminder. I sent around when to meet. So if you want to attend, just fill out your availability. Right now it's looking like Thursday is better for everybody, but I only have about 36 people. I think it's 65 response. So I just wanted to give you one more chance, and then I'll email that out either tonight or tomorrow morning.
Yes, some of you, that's great. Some of you, again, not coming from a science background or have never seen anatomy figures or never heard of neurophysiology, you're going to be overwhelmed by the terminology and everything. So it's important that you have some basic understanding of these things. For that reason, we have this crash course. It will be recorded, so if you cannot attend it, you could watch it later. But you should all, if it fits your schedule, attend it and ask questions. If it doesn't, then just watch the recording. It's really just to introduce you to the basics. And again, for it to also be a resource for you to go back to if you need to refresh your memory about certain basic concepts. Yeah, we'll schedule that. You'll get an email about it, you'll get a Zoom link about it, and then you could attend that. Next week, we'll continue on neuroanatomy and physiology. We'll talk about methods in cognitive neuroscience. We'll have added, again, some reading stuff. Kate also has added some extra reading material for those of you who are more interested in the clinical application of some of these topics. So if you're into that, you have the option of reading those. You're not going to be tested on any of these things. As I said, you'll only be tested on the lecture material. The following week, we'll talk about methodologies in cognitive neuroscience and neurocomputation. We'll have an exam one review session. Is this date something you guys decided?
Yeah.
Okay, great. So the date is that. The exam is the following week. So this is on the Thursday prior to the exam. And again, this is something you should all attend. They're going to walk you, if you have any questions about, it's important, as we get closer to that, I'll talk about it, but it's important that you work through the questions yourself and then come to the study session and talk about, and then ask your questions. You'll get a lot more out of it if you try it yourself first. It's true of a lot of things in life. Then we'll have exam one on September 17th, week 4. After the exam, on that week, we'll have a couple of tutorials on APA style and how to read a paper. This is all in anticipation of you actually reading a paper and doing your assignment. So, so the intention is to give you this material for you to worry about exam one for the first 3 weeks up till week 4, and then you switch your focus on reading papers and understanding how to read papers. I'll talk a little bit about it myself, but then this is where the assignment 1 comes in, and you'll walk through that. A couple of weeks later, we'll have a resubmission of assignment 1. This is to give you a chance, for those of you who've never done this, to get feedback from the TAs and then resubmit it after fixing whatever issues that they pointed out. This is, again, to give you an opportunity to improve your grade and your submission. A few weeks later, we'll have exam 2, assignment 2. Again, as we get closer to these, I'll explain them in more details. The last exam will be a week before the last day of classes. The reason being that I know you have other classes and they tend to have exams on the last day. Last couple of years, I think there was a final exam on the same day that I had it, so I wanted it to be on a different day. Also, it gives us a chance to wrap up, you could ask your questions. I could open it up like a forum for you to give feedback. We can also have time to do our course evaluations. And also answer any questions you have about writing your final paper, which will be due by the end of that week, by that Friday. After this, there are some boilerplate stuff about academic integrity and other resources that you could go to. Just check them out if they're relevant to you in terms of student services or those sorts of things. At some point here, you'll have to submit your final paper topic. So this is something that I would like you to start working with after you do your second assignment. Then your focus should shift to finding a good topic for your final paper. And then working on it. So you will check that topic with your TA. They'll guide you through narrowing it down to something more manageable. And then at that point, you can start working on it. But do this a few weeks before the actual due date. So it's just, you have time. As I said, the topic could be anything that interests you and it's relevant to a topic in the class. It can't be on anything. It has to be related to cognition. It has to be related to neuroscience and also something that you're interested in. Any questions about the syllabus? Is this all clear? Yeah. The exams are all in class. We'll print them out. We'll bring them here, and then the essay questions and the multiple choice questions, you'll just write them down here with pen and paper. Basically, yeah.
For the readings, are those to prepare for the next? Like, are those supposed to be done after?
No,
For the next time?
no, not necessarily. The textbook is stuff you could read before the class. The papers you could read after the class, if like further readings. But I would say the textbook you could read before the class. It would help you absorb the material better if you do it before the class. Yeah, they're not cumulative. So the material, you just have to focus on the material for those few weeks. It's easier to do it that way. Yeah. You don't have to worry about it too much. Questions?
The recordings will be on syllabus.
No, the recordings will be on Canvas. I will upload them to Canvas. And also I could share the link to them on Zoom. The thing with the Zoom links is that they expire after a month, but that's enough time because we'll have the exam before when it kind of expires. It works. So you could watch it online on the Zoom website, but also we'll put the recording on Canvas. Canvas. Everything goes through Canvas. We'll put the assignments on Canvas. We'll put the final paper submission on Canvas, and then everything goes through Canvas. Have a question? All right, so as I said, go to the syllabus. It has a lot more information that I covered today. The class is very heterogeneous. Like, so people who are accepted to this program, to the master's program, come from different backgrounds. And we are aware of that. It's not like we don't know that, I'm sure there are a lot of you from humanities background, they're from science background. Some people have done another master's somewhere else. So that heterogeneity is challenging. But it's like a normal distribution, right? There are a lot of people are in the middle, and then you have people who are on the extremes in terms of people who have a background or have done a master's, or maybe they've done a bachelor's in neuroscience. The course will come easier to those people in some ways. And if you're on the other extreme, the course may come harder to you. But the intention here is that we're not going to assume that you have any background, right? Having the crash course, having tutorials for reading papers, starting from the real basic. The idea is that if you're coming from any background, if you work hard and if you take it serious, you're going to do well. It's not a, it's really not that hard to get a good grade in this course. We have a lot of opportunities for you to learn the material and then to do well. Anything? Any questions? If not, we're going to move on. Anything you guys want to add or? All right. Okay, so I'm going to upload the lecture slides before the class, hopefully a few days before the class, so you can all, take a look at them and then, study them to some extent if that helps you. And then when you come to the class again, so you could absorb it better. Okay, neurophysiology. All right, so I am gonna use this construct that you see on the screen at times to just contextualize what we're learning. It is a simplification, but imagine an organization in the world, right? Us or even simpler organizations that are in the world and they're acting. You can imagine the relationship to the world in this perceptual cycle. Let's imagine that you have an environmental stimulus. A stimulus is just a fancy word for some input. It could be light, it could be sound, it could be a smell, molecules traveling in the world. Then this environmental stimulus is attended to, right? I'm not seeing everything. If I look at something, I could attend to it. And that's where the concept of attention comes. We'll talk about it. I'm just giving you an overview to contextualize something. I don't expect you to understand everything here. So when you're attending to something, what happens is that if I'm looking at a certain area, then the features, when the visual input from that area enters my eyes, right? If I'm looking this way, I'm not getting those inputs. So they're being attended. The stimulus basically reaches, the eye. It becomes neural activity in a process that is called transduction. It then gets processed through my background knowledge, and then it becomes perception. I'm perceiving that sign. And then I could recognize it. I could recognize the words, and then I could act upon it. If I was looking for a, let's say, the plug to turn on something, right? You could imagine how this cycle would be at play, right? So I'm looking for a plug, I'm going to look around, and then I find the plug. So this is the attention process. And then I could act upon it. I could turn on the switch or I could turn off the switch. You can imagine that this process plays out for a lot of organisms that act in the world. They are attending to different things in their environment. Then when they process it, they could act upon it. This is a simplification. A lot of time we're not, stuck in a cycle. A lot of time our actions are driven by some internal motivations or intrinsic motivations that are not captured by this. But people have simplified it, and a lot of time in science, you see these boundaries that are somewhat arbitrary. Like somebody does neurophysiology. What does it mean? It means that they're really interested in the connection between an environmental stimulus and the processing in the brain, right? So they are only interested in looking like, okay, this environmental stimulus like light how is it being processed in the brain? What kind of neural activity it has? All right, I have a slide for this, so I'm going to go to that before. So on a psychophysical level, it's really the study between the attended stimulus and your perception. So if I play a sound, right, and then I play another sound, and then ask you which one was louder, right? So I'm studying, what am I studying here? I'm studying the relationship between this stimulus in the environment, like the sound, and your perception. So I'm a psychophysicist at that point. If I'm studying neurophysiological level, I'm really interested to see what was the brain activity when you heard each of those sounds. Then I'm studying neurophysiological level. A lot of time when people study one of these levels, they really don't care much about other levels, meaning that when you study the relationship between, let's say, a pattern of light and neural activity in the brain, you could even ignore behavior. A lot of these experiments are done in anesthetized cats or anesthetized animals, meaning that the animal is not even awake. Well, what you're doing is that you're putting the animal to sleep, you're opening their eyes, then you're flashing different patterns of light, and they're looking at their brain activity. And it's really the relationship between these patterns of lights and that neural activity that you're interested in. You can imagine different patterns of lights, or if it's a face, or if it's an object, it could elicit different kind of response in the brain. So that's what's important in neurophysiology. Perceptual neuroscience is the relationship between brain activity and person's perception. So in that case, for example, with functional magnetic resonance imaging that we'll talk about, you could see that, well, what is the person perceiving? How is their perception is related to the neural activity? But these are just, again, I'm just introducing you to these things. So again, don't get scared by these terminology. I'm just saying, when you hear somebody studies the brain, they're usually studying it at a certain level. And people claim, okay, oh, I'm a neurophysiologist, or I'm a psychophysicist. So people kind of use these labels to broadly define their research area. So when we look at this macroscopic view of the brain, this is the body, the brain is in the skull, and you may or may not have seen these terminologies. When they try to talk about the brain, so this is the front, and this is the back of the brain. So these are the eyes. So the back of the brain is posterior. That kind of makes sense. You have the anterior area in the front, dorsal and ventral. I don't expect you to memorize any of these things. This slide is here to just let you know that you're not, I don't expect any of you to go memorize all these different terminologies about where the different brain areas are. But we'll get to a point where we talk about some specific brain areas and what they do. At that point, you're supposed to know it. But in general, I'm not going to ask you about these sorts of orientation and how the terminology works here. But if I talk about, let's say, inferior temporal cortex is important for object recognition, in that context, I would expect you to know those things when we talk about it. So we will use some of this terminology. We'll use, for example, ventral pathway, dorsal pathway. We'll talk about, we use this terminology, but it becomes much easier to remember them in the context of a specific brain areas and a specific cognitive behavior that we're interested in. So don't worry about it. Again, these are all to show you that you shouldn't worry about it. I don't know if it's working. So if we zoom in, right, the brain is in the skull. And if we zoom in into that brain, like any tissue, any living tissue, right, there are cells in there. And if it means anything to you, in the brain, in any small millimeter cube of the brain, there are 10,000 cells. The cells in the brain are specifically called neurons. And in this small area, if you could imagine it, there are around 3 kilometers, or about, is that 1.5, like, or almost 2 miles of wires. connecting these cells, right? The difference between the brain tissue and any other tissue in the body is that the brain tissues are connected to each other. The cells are connected to each other. The neurons are connected to each other, as opposed to the other tissues that the cells are basically next to each other. So that really differentiates the brain from the other tissues in the body. And for example, under a microscope, a typical brain cell or a neuron looks like this. There is a cell body. There's a cell body called soma, and then you have dendrites, and you have axon. There is great variability between different cells in the brain, between different neurons in the brain. But we're going to look at very specific cells, which are called pyramidal cells in the cortex. Those are the cells that we're going to generally care about in this course. So you have to only worry about these, but not all cells look like this. This is just a stereotypical pyramidal cell in the cortex. And we're going to use that to discuss anatomy of a cell. Let me skip this for now. Okay. So what does a neuron in the brain look like? Well, this is a schematic. But in this schematic, you could see the most important parts of the cell. There is a cell body. The cell body is like any other cell in the body, right? The cell has a certain machinery, which is very complex. If you had any biology course, you would know that. Where you have a nucleus, you have the DNA, and there is a lot of processes that is going on to sustain this cell and to burn energy, to receive energy, all sorts of things that are happening in that machinery. We're not going to care about any of that. What we care about is that the cell has certain branches, like a tree, that are called dendrites, that receive inputs from other cells. The cell also has an axon, with axon terminals at the end that are connecting to the other cells. So as I said, the most important quality of a cell in the brain, a neuron, compared to other cells in the body, is the fact that it could receive certain inputs that is connected to other cells, and then it could also submit that input to the other cells. You should know these body parts, the specific parts of a neuron, where the incoming inputs are through the dendrites. There's a cell body, and then that cell body is connected through axons and the axon terminals to the dendrites of the other cell. All right. So, for example, here, if you have a cell, a neuron that is sensitive to light, these are in the eye, basically. And when the light hits them, they could actually have a signal, this electric signal, and through the axon of this cell, it connects to the dendrites of the next cell, which has a cell body, and then that cell could also connect to other cells. So the working assumption here is that these cells in the brain are doing some sort of information processing. We're going to talk a lot about this computational perspective on the brain, right? We're going to talk about every aspects of cognition, recognition, attention, being a form of computation that is being performed by these neurons. And each neuron is, in a way, an information processing machine, that they're taking input from other cells, other neurons, or receptors that are connected to the world, and then it's sending that information to other cells. So this is our working assumption. As I said, we're not going to worry about the machinery inside of the cell that much, just a little bit. We're not going to worry too much about how a cell is sustained and stuff like that. All we care about is that the cell gets certain inputs, and through a mechanism computes something, and then sends that down to other cells. You could imagine, I'm going to use the computer metaphor a lot, right? That we're computing something, it's like a computer. Part of the reason is that when people are faced with complex systems, complex natural systems, we tend to use metaphors from artificial systems, right? There was a time that people thought that, for example, the brain was like a telephone system. Look how these, all these telephone systems are connected to each other. We have these hubs. When you call someone, it goes through the hub and then it connects you to the other place. There was a time they used maybe a steam engine to describe these sorts of computation. More recently, we use the computer metaphor, that the brain is computing something. But there are other metaphors that, have you heard other metaphors being used to describe the computation in the brain? Have you heard the brain is a computer metaphor? You'll get to know that a lot in this course, basically understanding what that means.
Does neuropsychology have theory of
Sure.
based on chemicals?
Yeah, like, I mean, explaining what theory of mind is. Is that what you mean? Like how you're able to assign agency to other people?
Yeah, like consciousness.
Yeah, well, if you have time by the end of the course, we'll talk about consciousness a little bit. Theory of mind doesn't necessarily need consciousness. You could assign agency to other people and kind of have an idea of what their motives are and stuff like that without talking about consciousness. But maybe if we get there, if by the end of the course, if we get there. So the way this course is organized, the first module, the first few weeks are going to be on low-level brain processes. We're going to talk about neurons and how they communicate. The second module is going to be on recognition, cognition, mid-level cognitive processes. And then the last module is going to be on higher level of stuff, memory, emotions, hopefully consciousness if we get there. But metaphor, speaking of metaphors, you could use social media as a metaphor. You could, each of us, imagine what it means if each of us act as a neuron in a social network. What does that mean? Can you imagine what that means? When you're on a social media, let's say Twitter or X, and you receive certain inputs, how do you receive those inputs? Yeah, so you get it from your connections, right? You are connected if you're following someone. Let's say it's a social media that you only see information from people you're connected to. So you get input from all these people that you are already connected to, and then you pass on that information to other people that are connected to you. Does that make sense? So each of you is an agent. You choose to act as an agent, as a neuron in this network, to pass on certain information. So you sometimes, let's say on Twitter, you see something and then you don't believe it. Then you see another person posting it and then another person posting it. And then let's say you choose to retweet that. What does that do? Well, you are computing something as an agent. You're saying this is worthy of being passed on to other people because it seems to rely on a lot of information that is coming from a bunch of people. So I could trust this, and it's strong enough that I could pass it on to the other people, right? So collectively, we are creating these movements on a social media. And that's not an individual thing, right? One individual doesn't create a movement. It's that collectively we're part of this movement, we're part of that movement. Emotional states, consciousness, right? These are meta, I don't know if that makes sense to you, but it's not on the individual cell. One cell is not conscious. One cell doesn't have theory of mind, but a collection of cells has a theory of mind or consciousness or emotion. Can you imagine for example, in going with the social media metaphor, a movement on a social media, right? A vibe, a trend, say, is a collection of things, right? If I you cannot reduce a trend to an individual, right? Trend is something that only takes place when a collection of people are moving in certain direction. Does that make sense to everyone what that means? Okay. So, but we're talking about individuals here. We're talking about individual units in a bigger social network of the brain, say. Each of them receives certain input, and then it passes on that information to other members of that social media, something like that. So the way it does that is that it has to have a way to receive a lot of input. And it receives these inputs through what you call dendrites. It computes whatever it needs to compute in this cell body. And that computation is really a summation of all these inputs. We're going to talk about that. As I used in my example, if you see something being shared by a lot of your friends, a lot of your connections, you are likely to pass that on to your own connections. So in a way, that summation happens. And then if you choose that, okay, this is good information, you pass it on to other members that you're connected to. Yeah, it kind of makes sense. Questions about this? We're going to go into a lot more details on this, but I just want you to go into it with a mental model of what it means to communicate. What does it mean for a cell to communicate in a bigger network of cells? It's good to have a mental model. I mean, the mental model is always a little wrong, but it's helpful. So let's see. Should we take a break? So we usually do like a 10-minute break in the middle, just for people to regroup. Let me pause this. Okay. So, as I said, we're going to understand the neuron as a computational unit. And that means a neuron has to have a way to integrate inputs coming from different sources, compute something, and then send the signal to other neurons. In biology, that means that everything that I just described has to happen through chemistry. Right? There's no other thing, right? The brain is a biological organ, and computing and signaling and all these things have a chemical process. So we're going to learn about that chemical process now. The cell neurons have a semi-permeable membrane. Permeable in a sense that they can allow things to go in and out of the cell. Semi in a sense that not everything can go in and out. There are rules to what can go in, what can go out, and we're going to look at what that means for the cell computation and signaling. So normally, when you have these membranes of the cell, and imagine this is inside of the cell, and these parts are outside of the cell, you have a lot of ions just traveling in the world, in this, in the brain. Sodium ion and chloride ions. Where have you seen this? Salt, right? Basically, if you dissolve salt in water, you're going to get positive sodium ions and negative chloride ions. And that salty taste that you feel when you have something too salty is basically the sodium, positive sodium ion interacting with your taste buds. So these ions are just around. Normally, there are a lot more negative ions in the cell than there are positive ones. So the net potential of a cell, imagine this is inside the cell, the net potential inside of a cell is negative when it's resting. So when it's not doing anything, there are a lot of negative ions just sitting inside of the cell. There are some positive ions, but there are far more negative ions than there are positive ions. So the net potential inside of a cell versus outside of a cell is around minus 70 millivolt. So when the cell gets excited, now this is where the part where you get a lot of input, excitatory input from its surrounding, and we'll talk about what that means. When the cell gets excited, these sodium ions that are outside of the cell rush inside of the cell. They go inside of the cell. Now, after a little bit it gets inside of the cell, then these gates open up. When the gate open up, then there is a rush of sodium inside of the cell. And because they're positive, they overpower basically the negative ions inside of the cell, and the potential inside of the cell could become really positive, plus 40 millivolts. This could only happen because there are these protein molecules that are gating this input and output. When the cell is resting, these gates make sure that there are a lot of negative ions inside of the cell. But when the inside of the cell gets a little positive, a lot of these gates are potential gates. So they suddenly open up, and because there are a lot of sodium outside of the cell, they kind of repel each other, they rush inside of the cell. And that's what makes it a semi-permeable surface, because it's just dependent on certain aspects, but things can go in and out of the cell usually. So what is the result of that? Well, when sodium rushes inside of the cell, it makes it really positive. And it's as positive as it can get at this point. Then, when it's really positive, the potassium cells inside of the cell rush outside. So these potassium cells only open up when the potential inside of the cell is really positive. These are also basically potential gated. So imagine a gate that for potassium that opens up only when the inside of the cell is really positive. So because potassium is also positive, rushing outside of the cell brings back the potential into the resting state. So if positive ions rush inside of the cell, they're going to increase the potential inside of the cell because it's going to be all positive. When the positive ions rush outside of the cell, they're going to decrease. So this process is called an action potential. This is how a cell communicates to the other cells. When this process happens, when the potential inside of a cell really goes up, and then it has to come down. So, and then after this happened, after the action potential has happened, there's a refractory period, because we have to go back to the starting point. But why is this not the starting point? So if the potential went up and we went down, why is this not the starting point? What else is supposed to happen? Louder? Yes, right? Because initially we said this is the resting state. The resting state is that there are a lot of sodium outside of the cell, a lot of potassium inside of the cell. But at this point, when we reach this point, there's a lot of potassium outside of the cell, a lot of sodium inside of the cell. The potential is the same as it was in the beginning, but not the ion distribution is not the same. So we have to have these gates where they're basically pumps, where they pump the sodium that is outside, the potassium that is outside of the cell back into the cell. And then the sodium that is inside of the cell back outside the cell. So you need that refractory period, as they call it, in order to actually go to the starting point. So to restore the resting level, actively exchanges so that, sodium and potassium using the sodium-potassium pump. So this process that I just described, that it has 4 steps. Let's go through the steps again. You're looking at the potential inside of the cell. Inside of the cell, it's negative because there are a lot of chloride versus the positive stuff that are sodium. And there are potassium too, but it's overpowering. The chloride, which is negative, is overpowering. It's very, so it's negative 70 millivolt. Then when the cell gets excited, sodium can come inside of the cell and potassium slowly leaves the cell. At some point when it reaches really high up, the potassium rushes out and then the cell membrane comes back and then the pumps do their thing and then they bring the cell into the initial state. This whole process is called an action potential. And this whole process, including refractory period, really takes only 1 millisecond. So a cell technically could do this 1,000 times every second. Cells don't usually fire that much, but some of them go a few hundred times a second. So a few hundred times a second, they explode in this way, and then they go back down. Explode in potential, they go back down. Now, having this firing rate, it's in some ways, it's amazing. This machinery is amazing that happens. And I don't have the demos here, but like the way these gates work and everything, you can imagine it's such a elegant machinery in order to do all of these things. At the same time, it's kind of limiting, right? A cell in this system can only fire 1,000 times a second. So it's kind of what it can signal is kind of limited. How does this compare to, for example, a clock on a computer? Like how often can a transistor in a computer change its state? What is the clock? How fast is the clock in a computer, like in a laptop? Every second? Like a clock of a computer. When you buy a CPU, for example, I don't know, you guys don't worry about those stuff anymore. But when you buy a CPU, at the time you cared what the gigahertz was. Like you really cared when it was an Intel and the clock was like on gigahertz level. You know what I mean? Like that means that they are 1,000,000 times kind of faster than this. Like a clock on a PC, like this laptop, the computation is like 1,000,000 times faster than a neuron. Because it's a much faster clock. And the clock in a PC doesn't have an equivalence here necessarily, but it's much faster. So that's why you could do all these things on a computer. You could compute something much faster. But yeah, I don't know if you guys have intuition on computers necessarily. Okay, so this process is called action potential. We're going to talk about what makes a cell, right? We haven't yet talked about what excites a cell. What makes it initially for the sodium to come inside the cell, right? We talked about how initially the sodium would have to come inside the cell for the potential to go up. But you could imagine already that this is part of the message passing. This is part of the process of the cell accepting these inputs and then sending a signal. So the cell getting excited, it is part of this process of its getting excited. Process of its getting input. Right. Now, another thing is that the cell doesn't just do this once in the soma. So it actually, when it integrates, by now you kind of know, it gets all these inputs from its dendrites, and then it wants to send the signal down its axon. So the process starts in the axon hillock, and you have an action potential here. And then the action potential travels the extent of the the axon, because some of these axons are really long. Imagine if you have to have an axon that comes from the brain, goes to the brainstem, and then it's supposed to control a muscle, right? It's a long distance. And even inside the brain, there are connections that go from one hemisphere to another hemisphere, and they're kind of long and like centimeters. And um, and in order for that action potential to actually reach the end, which is the we'll talk about the synapses at the end, in order to reach there, it cannot just have one action potential that travels the entire path. It has to have multiple ones. It means that at this junction, the axon hillock, which is the connected to the soma, that there is an increasing potentiation, and then this increasing potentiation kind of travels, and then it causes another action potential here. It causes another action potential here. And that's why you have these myelin sheaths covering the axon in order for it to not be permeable during this process. So the potential doesn't die down. It kind of stays up. And then it causes another action potential, it causes another action potential, and another, and then that's how it gets to the end. And then at the end, it could actually control the muscle or have some impact on another neuron. So the action potential starts at the hillock, the axon hillock, and then travels down through these propagating waves. When it reaches the very end of it, it releases what we call neurotransmitters. So the way one neuron communicates with another neuron is that it causes an action potential. The action potential travels down the axon, and then when it reaches the receiving cell, then it releases neurotransmitters. And these neurotransmitters are what really causes the activation in the receiving neurons to change. We're gonna talk about the details of this in the next few slides. But where have you heard neurotransmitters? Okay, yes. So when you heard those things, what were you think they were?
Like signals that, I'm sorry, I'm from Argentina.
Okay.
It's like a signal that makes you happy. Dopamine, serotonin, like when you're hanging out with a friend, where the cell gives that signal to the other cell, and it's okay.
Yep. Yeah.
Still,
So
sorry.
yeah, no, it's all good. We want you to participate. Don't worry about it. So these are what neurotransmitters. You mentioned a couple of them: dopamine, serotonin, others. So the way they are actually what cause communication. You need neurotransmitters in order for the cells to communicate to one another. So that's what their function is. We're going to talk about it in more detail. Do you have a question?
Yeah, so just to make sure if I understood. So the actual potential is the capacity to get the neuron get excited, and for example, the myelin is the speed of the information of that excitement goes to the terminal of the neuron, right?
Yeah.
And the neurotransmitter is like the language of the communication of those neurons.
Yeah, that's better. Yeah. Yeah. They go outside the membrane. So the brain is in a body of water. So every cell has so the cells are sitting in water practically. And it goes out into that solution, as you could imagine. It's just liquid. It goes into that liquid, and then it could come back into the cell. It's just sitting in water, basically. And the water, that's where all these ions are kind of floating around. Like neurotransmitters are floating around. Everything that is outside of the cell is floating around in that body of water. Yeah.
How many, would you go back to the propagating wave? How many different the neurotransmitter?
So in terms of,
1,000?
no, it's not in that order. No, it's much smaller. Yeah. These are really fast. Like the myelin really helps for it to be fast. So there are diseases, I don't know if you know of them, where when the myelin gets eroded or people lose the ability to create these myelins, it causes certain brain diseases that you may have heard of. Have you heard of? Yes. So that's one disease that is caused by the losing this myelin sheath, because it reduces the communication between brain areas, and also it reduces the communication between those brain areas and your muscles. So you lose the ability to move in a lot of those diseases. Because as I said, this becomes really important if you have a long distance connection. I don't know the exact relationship between how long it is and how many action potential you need, but that's a good question. I don't know the exact answer to. Yeah.
So, back to this concept of the brain sitting in water, when you're taking something like an SSRI or norepinephrine reuptake inhibitor, basically like those neurotransmitters are just sitting in this water.
So if you take the serotonin reuptake inhibitor, there's a double negative here, right? There's a reuptake of serotonin and you inhibit that. It means that you're letting serotonin just sit in this body, and that makes things a lot more exciting. So your brain is really excitable, and we'll talk about why. Yeah. Sure, we'll get there. Yeah, we'll talk about that a little more. We'll talk about neurotransmitter a little bit more. But you all are on the right track in terms of, as I said, when you even think about these diseases, like this computational view of the brain is important, not for just understanding cognition, but also understanding these diseases, right? There's something is not being computed properly, or what's being computed is not being communicated properly. All of those things can cause these different diseases, right? If you cannot, if cells cannot communicate with each other, you get all these depression symptoms. If the action potentials are dying out, you get some other diseases. So this computational view of the brain is also applicable to a lot of diseases as well. All right, so what happens when two cells are communicating? There is a gap between so let's go back to this. All right, so as I said here, there are dendrites. The dendrites are the receiving end of a cell. It goes through the cell body, and then whatever gets computed could be a summation of all the inputs that you have. It passes on through the axon, and then at the end, there are axon terminals. So naturally, the axon terminals from one neuron connects to the dendrites of the other neuron. When the axon terminal of one cell meet the dendrites of the other cell, this is called a synapse. So this is what a synapse is. So this is the axon terminal of one cell, and this is the dendrites of the other cell. This is the incoming, this is the outgoing. This is the presynaptic, this is the postsynaptic, right? The synapse is this gap. You have the cell that is coming with signal, so it's presynaptic, and then there's a cell on the receiving end, so it's postsynaptic, right? The terminology makes sense. This junction called a synapse. Now, if the action potential that you saw earlier is caused in the cell, and it happens to travel the axon and reach this point, then it can impact the postsynaptic neuron. The synapse is the gap between two neurons, typically between the terminal branches of one and the dendrites of another. Okay, so what happens at the synapse level? Well, When the action potential reaches the end of the axon, it triggers the release of neurotransmitters. As I said, anything in biology would have to happen through chemistry, right? There is no other way. If I have a biological machine, every mechanism in that biological machine is chemistry. So in this case, when the action potential travels the extent of the axon, it reaches the synapse, it releases neurotransmitters. The neurotransmitters can then go and bind to the postsynaptic dendrites. Now, can you imagine what kind of operations? So if these neurotransmitters are excitatory, what does that mean? If a presynaptic cell releases chemical compounds that are excitatory, what does it mean? Yeah. It means, yeah, exactly. It means that it's going to cause the next cell to be excitable. What does it mean to be excitable? It means that there's a good potential that that cell going to fire, that is potential inside of the cell going to go up. If the presynaptic cells releases chemicals that are inhibitory, neurotransmitters that are inhibitory, what does that mean? So what does it describe that? What does it mean when a cell is less likely? What does it mean for its potential? Yeah, so this, you are in this cell, you release inhibitory neurotransmitter, something like GABA. These GABA cells, molecules gonna go attach to the next cell. What can they do to make that cell less excitable? Yeah. Yeah. Yes, and what else, for example?
Stop the next cell from the uptake of the neurotransmitter.
So no, the reuptaking is a different operation. Let's think about how when a cell is inhibited. Yes.
There's like negative potential. So if that
Sure.
potential doesn't happen, so it stays negative.
It stays negative, or even it becomes more negative. How would it become more negative? Yeah. Yeah, that's what I'm asking. So what could cause a cell to become more negative? Yeah.
Sodium.
Sodium. So imagine a lot of sodium is outside of the cell.
But there's some in that diagram, there was some inside.
Yeah, a little bit inside, but think about what else? Potassium. So what other, so we have negative ions, right? Like chloride. So if I'm a neurotransmitter and I'm released from a cell and I'm binding to the next cell, what can I do to make that cell less excitable? What kind of ion channels can I impact? Chloride. What can I do with chloride? Well, I can open up the chloride and let chloride into the cell. Right? I could go there, open up some chloride gates, and then let chloride inside of the cell. What happens if I let chloride inside of the cell? What happens? So let's say the cell was minus 70 millivolt. What happens if I let chlorides in there? It becomes more negative. It becomes less likely to reach a threshold that it could explode, right? And you could imagine the opposite would be true if it was a positive neurotransmitter, if it was an excitatory neurotransmitter, where you're opening up ion gates that are positive and letting the positive signals go in there. Now, what happens if I open up, what happens if it's excitatory? If a neurotransmitter like serotonin is out there, what does it do to the postsynaptic cell? Yeah, but what does a neurotransmitter do? What did you say? Yeah, you said it. Okay. So it initially lets in positive ions in there, the potential goes a little higher, and then when it reaches a threshold, it opens up all the sodium channels, and then that just, pours into the cell. Does that make sense? It's a machinery. It's just a chemical machinery. So the cells can either inhibit each other or they can excite each other. So you can imagine it could start from light, right? I open my eyes, and we'll talk about the eye a lot in a few weeks. But I could open my eye, the light comes through, and it excites some cell. Those cells can excite some other cells. Those cells can excite some other cells, right? And there's this cascade of activation. You open your eyes, and the cascade of activation goes through your brain. Yes? What do you mean by opposite way? Okay. I'm not sure if I understand what opposite way means. So there is. Mm. Sure. I'm not sure. Hmm? Yeah. Yeah.
So I just wonder if how this,
So great question. I'm not the right person to answer that question though. You should go read on that. You should. I'm not familiar. If you guys want to pick on those questions, I'm not familiar with all the neurodivergent um, and the ways that cell communication or neuronal communication is impacted in each of them. But that's something you could read on. We're going to cover in this course mostly. Yeah.
I would just say that for that question, I mean, I don't know the ins and outs of each one, but I would expect that something like Asperger's versus ADD or ADHD, they have slightly different changes, probably not to the basic systems of what we're talking about here, but in the same way that like depression or anxiety, we have different levels of neurotransmitters sort of floating around available. These types of basic mechanisms impact different diagnoses in different ways. So for instance, ADD, ADHD, your attention systems tend to be impacted, and these basic systems underlie all of those diagnoses. It would be a really great final topic paper to like look into what each specific one is for each diagnosis, and maybe even differences between diagnoses, because that's a really interesting sort of physiological topic there.
Great. Yeah. Yeah.
different types of cells have different thresholds, or like when they would need to open a gate to release positive or negative, or are all cells same?
No, cells are very different from each other. There's great variability, or there's great variability in cell structure, the way, how many inputs they usually get, how many outputs they have. There's a lot of variability, and the thresholds are different. Yes. It really depends on how close the dendrite is, because the dendrites that we're showing a very a schematic here, but these dendrites have receiving synapses in all places. And it really even depends on how close this is to the soma. You can imagine if you get neurotransmitter impacting areas closer to the soma, it's more likely that it would fire than if it's not. So there is great variability. Yeah. All good? Yeah.
So are there certain neurons that are more like excitable versus mechanical?
So it depends on where the baseline is, right? There are neurons that have a higher baseline activity, right? A cell, even if you don't do anything to it, it's gonna fire once in a while, right? Some cells have a very low baseline activity. So generally, maybe it doesn't fire anything once every second, and then you give it an input and it goes crazy. Some cells have a much higher baseline activity. So they maybe, have 10, 15 spikes. These are also called the spikes per second. And then you give them an input, and then they also, could be really active. So it really depends on what it means to be more or less excitable is in the eye of the beholder. Yeah. Yeah. Sure.
Structure, cell body, thing, or person growing to
Yeah. You work out. Yeah. That's it. It means having a healthy lifestyle. All that, whatever that carries, you could be physically. Previous what? Oh yeah, I think so. I think a lot of things carry forward, I suppose. Um, Again, this is a deeper question than one I would be able to answer, but I think no, it's okay. Um, I think I thought initially you were asking for an individual, right? So there are there are a lot of this stuff, there are a lot of studies showing that a healthy lifestyle promotes brain health in so many different ways. You could create new cells, you have better connections between those cells. It's like a um, whether you would carry it forward to a child or something. What was you asking? The You could use that. This is not a politically charged environment. You could use these words. We don't. Yeah. Yeah. I mean, I don't think. Yeah, I don't think it can hurt to work out, but I don't know the answer to the question of whether how much of this gets carried forward to a child, like if you're carrying a child. I don't know the answer to that, but on that note, this is a safe environment. You're in a classroom, so you could say even the wrong thing. You could use the wrong term. It's not a big deal. Just, yeah, you could just say, yeah. You don't have to, conform to some societal thing about political correctness here. Um, so I wanted you to hear what a cell sounds like. So you could potentially lower an electrode and listen to a cell. You could put an electrode right next to a cell and listen to this action potential going up and down, right? So like like when you make music a lot, right? If you do, you know that there are amplifiers and you could just have a signal, plug it into an amplifier, and then listen to it. So you could do that with a cell too. And a lot of early research in brain sciences were done this way. So this is a very this is a very seminal work. and these, we'll talk about this work a lot later in a few weeks. Hubel and Wiesel, they did a lot of these experiments where they showed pattern of lights to anesthetize cats. So these are cats that are not awake. Cats have a cortex, like visual cortex, that is very similar to humans. So that makes them a good model animal to study. So you put an electrode in the back of the brain. This is where the visual cortex is. We'll talk about it. The visual input goes through the brain, goes in the back. So if you put a cell, and this is just one cell among billions. So the sound you hear, it's really a cell firing. This is an action potential of a cell. So each dot that you hear back to back, that's just the sound of a cell firing. So for example, this cell fires when it sees this pattern of light in the environment. We'll see more of this video later, but just to give you an intuition of what an action potential is. So this is one cell receiving inputs from a lot of other cells, deciding that, okay, this is an action potential, and then it's passing it on to the next. There's a hierarchy, there's a cascade where the information gets carried forward in the brain. So cells are either excitatory or inhibitory. And what defines a cell to be either excitatory or inhibitory? You're close?
The concentration is more positive.
No. What makes a cell to be excitatory to other cells or inhibitory? When we say a cell is an excitatory cell or an inhibitory cell, it's in relation to other cells. What makes a cell an excitatory cell? Yeah? Yeah, so, yeah.
Not like above 70?
No. So, no, I'm saying a cell is excitatory when it excites other cells, right? The terminology goes this way. So if a cell is excitatory, when we call a cell, oh, this is an excitatory cell, it means that it excites other cells. No. Okay, what does that mean about that cell? What does it mean about this cell? Hmm? No, what is, go ahead.
Like certain cells have different roles.
Different roles? Sure. But there's something about a physiology of this cell. Yeah.
It's where it's close to threshold.
No, I'm not talking about, go ahead, sorry. No, so what I'm talking about, I'm saying this cell is excitatory. It means that it excites the postsynaptic cells, right? That's when we call a cell excitatory, this is not that this cell is excited. Those are two different. Yeah. It excites other cells. What does it mean about this cell? What is the defining characteristic? What is the biological property or chemical property of this cell?
Fire natural.
Okay. Well, what is the chemical property of this cell? Exactly, right. So what does it mean for this cell if it's excitatory? It releases. Exactly. So when a cell is excitatory, it means that it releases neurotransmitters that are excitatory. If a cell only releases serotonin neurotransmitters, its effect on its environment is always excitatory. And a cell is either excitatory or inhibitory. It means that it cannot change roles. So if a cell only has GABA neurotransmitters inside of it in its synapses and only releases GABA into the synaptic cleft, it means that it's an inhibitory cell.
On a chemical level, what makes cell inhibitory or excitatory?
Just the biology of the cell. Just the cell in its biology is an excitatory.
In the textbook, perhaps, like for a detailed explanation of that, what the biology is.
Most likely, yes. You could read on that if you're interested. But like any cell, right? What makes a cell a skin cell versus a cell that is, you know, a vein, or you see what I mean? Cells take on different roles depending on their biology and the way that the genes are expressed inside of them, depending on where they are.
The location of the cell also have the indication on what type of transmitter is sent out.
Not necessarily. The excitatory and inhibitory cells are next to each other usually. They're just all together. For example. Yeah. So the terminology, so I see the confusion. So a cell could get excited, which means that it could fire action potential. That's not the same thing to say the cell is an excitatory cell. When you say it's an excitatory cell, it means that it excites other cells. That its job, when it fires an action potential, the effect on postsynaptic cells is always excitatory. That's what it means. If it's an inhibitory cell, it means that the effect on the postsynaptic cell is always inhibitory. And that has only to do with the type of neurotransmitters it releases. Because both of them are firing, right? An inhibitory cell fires, releases GABA, an excitatory cell fires, releases serotonin or glutamate. And those things excite or inhibit depending on, but the behavior of the cell is the same, it's firing. It's the opposite, but there are also other neurotransmitters. Yes. Yes. Just the type of neurotransmitters they release. So, as I said, a cell can integrate inputs from all sorts of cells. So a cell, a typical pyramidal cell can get connections from 1,000 different pyramidal cells and other cells. So its dendrites, as I'm showing it here, its dendrites could have 1,000 synapses coming from different cells. Its axon can go and connect to 1,000 other cells through 1,000 synapses, right? So it's getting input from 1,000 different cells. It's sending output to 1,000 other cells. So what it does is that it combines all those information. Now you know some of those synapses are excitatory, some of them are inhibitory. So if the cell only gets input from the excitatory synapses, it's likely that its firing rate goes up. If it only gets input from inhibitory, it means that its firing rate could go down. But in reality, every cell gets a lot of different excitatory and a lot of different inhibitory, and then they balance each other out. And depending on which one is stronger, the cell firing either goes up or down. Yeah.
So with antipsychotics, the way they work is they basically pinch off the dopamine receptors. So what I'm wondering is basically when you start an action potential, let's say you've got a neuron firing a bunch of dopamine, the dopamine receptors in the next cell will take in that dopamine, that cell will then fire more dopamine or fire more dopamine, right? So basically I'm just trying to understand the difference between the reuptake inhibitor and the way the antipsychotics work. So yes, maybe there's dopamine that remains in the synapse, but you're pinching off the receptor, and so that next cell doesn't produce dopamine. So you've got a finite amount of dopamine that's ultimately produced.
Dopamine is generated somewhere else, right? These neurotransmitters are not generated by these cells necessarily, right? These are generated somewhere else. And then these cells tend to pick them up and then release them. But there is a whole like the neurotransmitter system that generates these cells. I don't know. Again, I'm not a clinical psychologist. Yes.
Like for example, sodium entered left.
Mm-hmm.
And I'm a little bit confused. The difference between these states and the neurotransmitters. I wonder if you could maybe speak to that a little bit.
Sure. So the neurotransmitters impact those gates. They just impact those gates. So you could, yeah, some of the gates, so the gates are impacted in different ways. Some of them are voltage gates, meaning that when the voltage goes at a certain level, these gates open up. So this is what happens when we're here. Let me go. So imagine this goes up a little bit, and Kate's gonna give you a more detailed explanation of this. I don't have time. But when the voltage goes up at a certain level, those voltage gates open up. But initially, the gates open up because of the neurotransmitters. So the neurotransmitters impact these gates. Yeah?
So just to clarify, would we see a major difference in the different type of excitable and neuron? Are the gates specific to which cell?
Are the gates specific, excitable? No, not necessarily. No, not necessarily. So again, the terminology that I know we're out of time, but think about this terminology of when something is excitatory, it's about what impact it has on postsynaptic cells. When something is excited, it's a claim about what's its firing rate. So that's different. Those are two different things. Those are orthogonal. Don't confuse the two. We'll talk about this slide more next time, but this is the last slide here. I'll see you all next week. Sure.